Okay, let me share my screen. Okay, here. This one. Okay, guys, let's start today's lecture. So today is the second day of an antiviral drug. So as usual, we are going to solve several quiz problems together, and we move on. Okay, be ready for Kahoot. Hey, when I play this Kahoot, do you hear the music too?
No.
No.
No music sound?
Yes, no music sound.
This music sound. Do you hear that?
No.
No.
You
Professor,
don't hear music sound?
no. We cannot hear your music sound.
Something wrong. Right now, you don't hear any music sound?
Yes.
I think that's because you didn't share the computer sound.
Huh, how can I do it? Yeah, I was wondering whether how I can do it. Without the music, this is not fun.
Right? When you share your screen, there is a box in the left below corner.
When I do the screen sharing, where can I do it?
Click share screen, and there is a box at the left below corner of the of that window.
Ah,
And
yeah.
Yeah.
I see that. Now, do you hear the sound?
Yes.
Yes.
Ah, shit. Yeah, without this sound, this is not fun. Okay, got it. Okay, we have 5 of you. Who's not here?
Professor, I think the computer volume is too high and I can't hear Professor's voice.
You cannot hear my voice?
Yes.
I can hear it, but it's not like it gets muted because of the computer stuff.
Are you saying that you hear the music but you don't hear my voice?
No, I can hear both, but because the computer sound is big, your voice gets relatively lower.
Okay, let me turn off the computer sound then, or let me try to reduce the computer sound. Is it better? No, you don't hear my voice either?
Yeah, I can hear it now.
Okay, let me try to speak loud.
It's not improving. The sound of the computer is too loud.
Now do you hear me? Not good? Okay, let me turn off the computer sound. Nothing is perfect. Okay, turn off the computer sound. Now do 1 second. Let me see. Do you hear my voice?
Yes.
Yes.
Okay, then let's go with this. Okay, everybody's here. Sejin is here. Okay, let's just start. Yeah, nothing is perfect. Ha. Who is this lipid guy? Are you kidding? 이거 너무하는 거 아니야? The main 3 components of life forms on our the planet is protein, carbohydrate, and lipid. This is just checking the base. Okay, you don't need to tell me now, but I can check later who is this yellow guy here. Next problem is a real problem. Okay, perfect answer. Problem is I don't have that structure anymore. I just showed you adenosine monophosphate, and let's imagine there's a structure here. There was 5-membered ring carbohydrate in the middle, and there was a one group here. What is this group here?
Phosphate.
You don't
Phosphate.
remember? There was a phosphate group was there. So there was a one phosphate and one carbohydrate in the middle. And then there was another structure here. That structure either the purine and pyrimidine, which has a multiple nitrogen, which we call base. So this is a building block of genetic material. And this is genetic material. Interestingly, our 3 major components of our body is a protein, carbohydrate, lipid. But the key genetic information material that is composed of not protein. It is composed of a carbohydrate and connected by phosphate, and the real information is stored in base. Okay, it's a quite unique structure it has. So for now, we know the DNA is our genetic information, but just 1 century ago, it was a big debate. Nobody knew exactly. DNA is a relatively boring molecule. It has only 4 building blocks. And protein has 20 building blocks of amino acids. So protein is a much more complicated and also sophisticated structure. So when the scientists thought about the genetic material, the first candidate was always a protein. Protein may be transferred to our offsprings, carrying the information of ourselves. That was, common belief for a long time. So that's why 1950, Watson and Crick's double helix structure of DNA, and then the proof of the genetic information is in DNA is real revolution. Okay, so now this question is obvious to you, but protein was always a strong candidate of a genetic information material. Actually, maybe 20 years ago, there was another Nobel Prize was given to this topic. Protein can be genetic information in terms of a prion, the mad cow disease material. Still, there is some debate that still partial genetic information can be stored in proteins. So maybe we have a chance to talk about it later. Anyway, I'm just showing you the basic building block of genetic information. Now, let's look at the next question. Wow, quite broad. I asked active building block, not just the building blocks. So this question we are going to talk about later in the main course. Okay, so as I worried, some of you were not familiar with this term. Transcription. What is the term in Korean?
전사.
전사. That is copying from DNA to make RNA. This is a transcription. And 2 of you answered RNA to protein. What do you call this process?
Translation.
Translation. Okay. This is a transcription, and this is a translation. So the central dogma of biology, the modern biology, most of those life forms, information flows happens from DNA to RNA. And then by reading RNA information, we make a protein. That is central dogma of the the current biology. But sometimes there is a different function, different the process is known. Utilizing RNA information, we can make DNA. This process, what do you call?
Reverse transcription.
This is called as a reverse transcription. Reverse means it's opposite. Transcription is the same word from here. So normal process is utilizing DNA as a template. And then by reading this sequence, we make RNA. This is a transcription. Then if it is an opposite approach, then we call this as reverse transcription. Okay. Now, if we have a DNA, by reading the DNA information and synthesize DNA, this process, what would you like to call?
DNA replication.
This is called as a replication. Okay, replication means make same copy. So this means copy. Utilizing DNA, make DNA. This process we call replication. Okay. Now, if you think about the enzymes involved for each cases, now in this case, product is a DNA, right? So in this case, the enzyme involved in this process is called the DNA polymerase. Polymerase means polymer making enzyme, okay? This is a DNA polymerase. Then what do you call here? DNA to RNA. I gave you
factor.
RNA polymerase.
It can be RNA polymerase, right? Because it makes RNA, okay? Now, in this case, what would you like to call? As a class, the product is the DNA. This should be also a kind of a DNA polymerase, but this is a different kind of the polymerase because template is different. By reading RNA and then synthesize the DNA, this DNA polymerase should be different from this polymerase. Okay, so that's why this process is called reverse transcription, that we gave separate name for this DNA polymerase as
Reverse transcriptase.
reverse transcriptase. Okay, transcriptase is a Korean dialect. Okay, so transcriptase we pronounce. So this is reverse transcription, and the enzyme selectively doing this is the reverse transcriptase. In human body, we don't need this process. That's why we don't have that enzyme. Okay, this enzyme is only belonging to RNA virus, some of the RNA virus, and they need to make DNA utilizing RNA information, and then insert this DNA into our genome. Okay, so that's why if the virus has this enzyme, it can be good target, something which is not existing in our body, only virus has. Then if you can selectively attack this target, then that can be a good target for the virus to attack. Okay. Now move on. Okay, question may be a little tricky. Now, virus, depending on it is DNA virus or the RNA virus, viral DNA synthesis is necessary to make their copy of their genome. And if they need to make protein, virus also has several genes. And those genes are carrying the information to make a protein. If they want to make their own function or their coat protein, capsid protein, they need to synthesize viral proteins. Some of them is active enzyme, like a reverse transcriptase or proteases. Some of them is their structural protein, like a coat protein. So protein synthesis is also necessary. RNA synthesis, depending on whether it's RNA virus or transcription from DNA to RNA, this is also necessary. Now here, viral lipid. like an envelope of a virus. The key point here is maybe there is no specific lipid structure, only specific to virus. If you remember those exit of a virus particle, they make capsid complex with genetic information, and then they come out from the cell, but the cell is encapsulated by cell membrane structure. Means they can steal the lipid structure from host cells. They may not need to synthesize those specific viral lipid themselves. Okay, so this is the key point here. So other information that they needed to make and synthesize to make their copies and also to make the protective structures kind of thing. Okay, today we have just a 5 problem here. Now let's move on. Oops. Okay. Now the first target, the talk is acyclovir. Actually, this one was prepared by Sang-an, and I didn't give the chance to Sang-an for presentation for this because this acyclovir is actually introduced in the video material we have studied, which was developed by the women scientists in New York. And the quick summary of this. Now, this is a building block of DNA. Am I right?
Yes.
One wrong answer and the others didn't give an answer.
No.
I said that this is a wrong answer. No, because?
That,
No phosphate.
yes. That carbohydrate is not DNAs.
Okay, this is for
RNA.
Okay, this is for RNA. So we didn't learn this for details, so don't worry. Just I just checked whether you know that or not. So this is one building block of RNA, and then RNA's the core structure is this structure is so-called ribose. This is a 5 carbon sugar, 1, 2, 3, 4, 5 carbon sugar. The regular carbohydrate is a glucose. Glucose is a 6 carbon sugar. This one is a 5 membered ring and 5 carbon sugar. And connection of this guy is one of you said there is no phosphate. Yes, phosphate is missing. We need to have a phosphate linkage here and then connect it to next the unit of this up here. And then that connection happens this position to this position. Okay, connection to this way. So we give the number from here 1, 2, 3, 4, 5. So the connection happens between 3 positions. Actually, this is the real numbering. This one we call 1 prime, 2 prime, 3 prime, something like that. So 3 prime to 5 prime connection utilizing phosphate. That is the building block. Okay. So the 3 unit is 1 phosphate we need here, carbohydrate, and base structures. Base structure, in 2 weeks, we are going to learn it more in detail, but base structure, we have 4 of them, ATCG, you may have heard, okay? Now, connection happens here, and then acyclovir is a very similar structure to these building blocks. This base structure is exactly the same with the guanosine, and the only difference is this sugar structure is missing some part over here. So most important thing here is ribose has this hydroxyl group. DNA do not have this hydroxyl group. That's why it is called as a deoxyribose. That's why we call it DNA, not RNA, okay? Now connection happens only through here. So it doesn't matter whether you have this hydroxyl or not, okay? Now if you look at this structure, now this hydroxyl is there, but there is no 3' hydroxyl. So that's why if this is incorporated into the synthesis of RNA or DNA, now the next step synthesis is blocked because it doesn't have this linkage structure. Okay. So the idea here, the idea is acyclovir is the drug which can interfere with a regular DNA synthesis or RNA synthesis. Okay, that was the original idea. There is one more very critical idea here is, if you just remove this, then there are many polymerases. We have our own DNA polymerase, we have an RNA polymerase, and then how can this compound, this drug, acyclovir, can distinguish only virus RNA polymerase or the DNA polymerase? That is a key question, right? So the idea was introduced in video clip. You already have heard, but if I summarize it here, RNA has this specific kinase. To make this building block active, so we need only one phosphate here, but this one phosphate is not energetic enough to connect this motif to next hydroxyl group. You need to have this extra 2-phosphate. Now these 2-phosphate, the pyrophosphate, they provide enough energy to allow the linkage of this phosphate to the next one. Okay, so starting point here is, just this guanosine is the starting point. This can go inside the cell because it is not very hydrophilic. It's reasonably hydrophilic, it can go in. And then to make this building block ready for coupling, we need to add 1-phosphate and 2-phosphate and 3-phosphate. 2 is not enough. So previous question in the quiz, this is the G. I gave you A as an example, but G, this is guanosine. If you have 1-phosphate here, you call it GMP, guanosine monophosphate. If it's a 2-phosphate, you call it GDP, guanosine diphosphate. If you have one more, 3, then you call it GTP, that's a guanosine triphosphate. Only 3-phosphate loaded building block, this is active building block. Okay, that was a question in the quiz. Now, key idea for the selectivity here is only virus kinase. The first kinase is the enzyme which can transfer the phosphate, put phosphate onto the substrate. That's a kinase. This enzyme is only available in virus. Only virus make this enzyme, and virus can add first phosphate here, and then second phosphate, third phosphate addition can be happened by host enzymes. That's why if this compound goes into virus infected cell, then first step can happen. Okay, if the cell is not infected by the virus, then they don't have this enzyme. That's why the first step of phosphorylation does not happen. That's why non-infected cells are not incorporated this into DNA synthesis. Only virus infected cell, they initiate the first step, and then this can go further to the next step. Now this becomes active. This goes into DNA synthesis. This goes into DNA synthesis and incorporated here, but it doesn't have a 3' hydroxyl group. Now next step cannot happen. Do you follow me? So this selectivity explanation was given in the video. I believe you understood that part, but just to make sure, I just used the science presentation material just to remind you of this process. Okay, this process is also connected or related to other presentations for today, so this is good to know. Okay, based on this, now this is the drug we already have seen. Look at the name, acyclovir. And then I chose 3 presentations: Toyun's sofosbuvir, Sejin's remdesivir. And actually, the Yonghwan also gave the one very similar drug to this guy, but Yonghwan's selection was for HBV. This is for HCV. HBV is a B-type hepatitis B형 간염, and this is for C형 간염. The mechanism is quite similar to each other, but this drug was so successful, and this made maybe 100 times more money than HBV drug. Okay, and if it's a very similar mechanism, then you may not need to hear both of them. So that's why I selected HCV as the drug. And this drug was developed by now the famous the company Gilead. And Seijin chose this drug. This drug is also chosen. This was also developed by the Gilead, the same company. They share some common mechanism of this, but target is different. This compound is for HCV virus, very, very successful. HCV was a very difficult the disease to treat for a long time. This drug just was introduced maybe 2-3 years ago. And it's almost completely washed out the whole the disease. It has more than 99% of a therapeutic effect. So people believe that HCV we conquered. So 30-40 years ago, we believed that we conquered the 천연두. Now this may be second the disease we conquered very successfully. HBV, B형 간염, still we don't have a good the drug, but the more difficult disease we believe, HCV is conquered by this drug. Okay, that will be quite interesting story. This remdesivir, this is another interesting drug because this has been developed for different virus, but now this is claimed as to use against coronavirus right now. That's why this can be very timely the introduction of this. And Dong selected Avigan. This is also developed for other virus, but Dong will tell about the story. These two, you may have heard the name recently in the newspaper. Okay. Takjun selected a very interesting antiviral drug too, but Takjun's selection was an antibody as a drug. So antibody we will manage later in details. So I didn't select that part. So this is the reason how I chose these drugs for now. Now comparing to previous the acyclovir, and then if you look at these two drugs' name, there is some common part in the name. Did you figure out? In the name, there's some common the part. Which part is common?
VIR.
Here.
This is ending with the VIR, right? So this is systematic the naming of drugs. This naming is not randomly given by the company. There is international association which designate specific the name like this, and then company can give their own name, so-called the trade name. Okay, then you can see this part here, VIR. Now you can guess what it means, right? It means drug for virus.
Virus.
Okay, so this is the VIR is a virus. Another case you may see the later, MAB, you may see. Something is finished with the MAB. MAB, AB is antibody, M is a monoclonal. So if you see any compound name as a MAB, that's antibody drug. Okay, at least some of them has some clue what it means. Avigan, this may not be the systematic name. Maybe this is maybe company's trade name, or it doesn't have a systematic international other name yet, maybe. Okay, so based on this, now let's move to the presentation. So first presenter is Doyeon. Doyeon, would you share your slide?
Okay.
Okay, we can see.
May I start?
Sure.
Today, my topic is mechanism of soposbuvir. And let's start. Contents are three, that why I choose this antiviral drug and development of antiviral drug for HCV, C형 간염. And lastly, mechanism of surface behavior. Why I choose this topic? Because surface behavior is main product of number one product of Gilead Science, who the bestseller company. So I choose it. And its development. Let me introduce the HCV virus drug development. First generation antiviral drug for HCV are interferon alpha and ribavirin. Interferon alpha is about interferon, and ribavirin is also like soposbuvir, and I will say about later. And these drugs have side effects such as anemia, flu-like symptoms, mental disorders, etc. And more than half of patients are not completely eradicated from the virus, even if treated. Second generation antiviral drug is also known as direct act antivirus, DAAs. They inhibit NS3, which is protease of HCV. The cure rate was rising to about 70%, but it still has side effects. And topic, sofosbuvir is third generation drug. Is a competitive inhibitor of RNA polymerase as ribavirin. And it conquer the HCV virus. So what's the difference between ribavirin and sofosbuvir? The previous problem is about phosphoric acid. To activate position, it must have 3 phosphoric acid, but ribavirin has just one phosphoric acid, just no phosphoric acid, so it go into the cell. It must have a long time to activate. But if drug did phosphorylation before using to make short time to activate, it has low transmittance because lipid is hydrophobic, but this drug is hydrophilic. Mechanism of sofosbuvir is using prodrug. Prodrug is previous chemical structure, and then add a more complicated chemical structure, such as pennyl, and so they did remove negative charge of phosphate acid, so it changed to hydrophobic, and it can activate faster than before. And thank you.
Okay, can you go back to the first page before I invite the questions from you guys? Second page, small point here. Doyun said why I choose this antiviral drug. In this case, the choice of Doyun was made now. Doyun, when did you choose this anti-drug?
Now.
Did you choose it now or the?
2 days ago.
2 days ago, right? So that's a past the situation. So you may say why I chose this topic. Okay, so it's a minor thing, but that's better. Let's move on. Next. Go back. Actually, in the beginning, the drug, the Gilead, the company's the drug was actually quite expensive. It was almost one set of treatment was about 1,000만 won. So that was quite expensive in the beginning. But very quickly, we have a very strong health insurance system in Korea. So it was maybe the 2,000만 won in other countries like in US or in Singapore. But in Korea, they made it quite quickly, about the 100만 won, 200만 won 수준 at the level. So it became very popular very quickly, and price was not a big issue. Next. And the effect was really good. That's why it wiped out the HCV almost completely now. Interferon alpha is an immune activator. It's kind of the signaling molecule in our body. You may have heard recently cytokine storm. Cytokine is a collecting of all of these interferon, interleukin kind of immune signaling molecules. And interferon alpha is one of them. The idea here is interferon alpha can boost our immune system and attack the virus. Of course, it may have a lot of side effects because overactivation of the immune system is also a problem. Next page. Next. Oh, sorry, stop, go up. Another enzyme name here, second line, inhibit the NS3, which is a protease. Protease is another important enzyme. This enzyme's function is a cleave protein structure. Cleave specific site of a protein that generate different useful fragment. So virus usually they synthesize one big protein. It's combined several enzymes together, and this protease should cleave in the right position to make them separate. Then they can be activated. Okay, so that's why protease is also another important target of a viral drug attack. Okay, move on. Next. Next. Okay, this is a key page for the presentation. Now, do you have any questions? No question? Everything is clear?
I have a question.
I have a question.
Don't first.
Really? Well, I'm actually not asking a question because I'm just actually quite confused what the exact diagram looks like. So I see many chemicals, but I don't know what inhibits what and what transforms into other things. So I would like to have one more explanation about it.
Okay,
This is my question too.
Doyun, would you like to try to explain this the slide again?
Okay. This one is just nucleoside and add up to add phosphate exit. It's been it must be have a long time. But if one phosphate exceed here, it to add up two phosphate to activated triphosphate much faster than zero phosphate to add two phosphate exit. So suppose we had one phosphated exit and phenyl. To first reason is to fast activation, and reason two is remove the negative charge of phosphated exit, so it changed to hydrophobic, and it can penetrate lipid 껍질 easier. So this slide says that why Sophos-Bouvier mechanism is perfect.
Okay, let me help you a little. So here, maybe original idea can be maybe first nucleoside, the structure on the left side. I don't have the cursor, the function. You don't see my cursor, right? This page is shared by Doyun, so you may not see my cursor. Left side, the nucleoside, it can be potentially antiviral drug. Now, if you use the left side, the nucleoside compound into the cell, then usually the first step of a phosphate addition is very slow. That's why before it becomes activated to move to second, third phosphorylation happens, then it takes a too long time. So that's why the effect of a drug is very slow. Then your second choice, maybe, in the middle of the compound, nucleotide monophosphate, it can be good drug candidate. Then it's fast to move to the next step, and then blocking those DNA or RNA synthesis. That's something you can expect. Now, problem is, the second compound, nucleotide monophosphate, it has a phosphate group. Phosphate has a two negatively charged. If it has a two negative charge, then the hydrophilicity is very high. On Tuesday, we talked about the absorption of the drug compound into the cell. We compared the two compounds, right? Relenza and Tamiflu. Relenza was too hydrophilic. That's why it could not penetrate the cell membrane easily. And Tamiflu became simpler and more hydrophobic, and it could penetrate the cell membrane much easier than Relenza. Now, that situation happens here. Because of this phosphate, not only single negative charges like a carboxylate, it's a double negatively charged. This is too hydrophilic. And this compound has a very low chance to go inside the cell, right? So to overcome this problem, the idea is they use kind of a protected phosphate compound. That's the Bouvier compound, which has a phosphate, but that negatively charged is blocked by phenyl group and also another some functional group here. They have protected this phosphate group with this group, this functional group. That's why the whole molecule does not have any charges. Okay, now this molecule can go inside the cell. And other enzymes inside the cell, in this case, the mainly hydrolase the enzymes, they clip off this protecting group, they remove them, and they generate the active component in the middle, nucleotide monophosphate compound as active drug component. Okay, so the real active component is in the middle, that phosphate the containing compound, but added compound form is kind of a protected drug. That kind of drug we call pro-drug. In this case, the pro does not mean that the professional, it means before, okay? It's not, itself is not drug, but it's kind of a precursor of drug. That's a pro-drug. So by utilizing this pro-drug concept, and they provide this active component inside the cell, and then this compound can go to the next step very quickly. Then it can make a diphosphate, triphosphate, and it becomes activated. Okay, so this kind of approach was quite a unique approach of Gilead. And then the company was originally a very small company, but with this success of this drug, they obtained a lot of money and also fame. And they applied this concept to different drug development. Okay, so historically, this is quite the unique and also quite successful story. Youngwon's HBV, the drug, also utilized very similar concept here. But HBV drug was not so successful, but this drug was great success. That's why this is survived the #1 drug in their company. And now we move to the next talk. Then we will see how this kind of a concept has been applied to another virus drugs. Okay,
Professor.
Yeah, go ahead.
I don't understand what is the role of soposbuvir when this drug go into the cell.
Okay, if this goes inside the cell, then other enzymes inside the cell, they cleave off the phenyl group and also those N connected the peptide group, they remove them. That deprotection is active process inside the cell. Many enzymes can do this hydrolysis. Then this generate the active component in the middle, nucleotide monophosphate. Then and this compound can be further phosphorylated 2 times, and then it make a triphosphate, the right side active triphosphate. And this compound can incorporate into RNA synthesis. Then when the virus RNA was synthesized, this compound goes in and then connected. And even though it has a 3-position hydroxyl group, the other side of the compound structure is not exactly the same. That's why the RNA structure cannot be synthesized for long. That's why the RNA synthesis is blocked. That RNA synthesis is unique in HCV. That's why the virus cannot survive.
Okay, thank you.
Okay.
Actually, I have more detailed answer against your question in my presentation.
Okay, go ahead. Okay. So we can hear some more story from Sejin, I think. Okay, Daeun, could you take out your slide? Okay. Let's move on. Okay, perfect. Go ahead.
All right. Yeah, hello, I'm Sejin. Today I want to talk about one of well-known antiviral drug, remdesivir. I believe some of you already heard about this in connection with COVID-19. So let's take a look at how this can solve the disease. Remdesivir is another antiviral drug in the class of nucleotide analogs like the previous drug, which means the drug mimics a structure of nucleotide containing sugar, phosphate group, whatever. Remdesivir is in the case of remdesivir, it is an adenosine analog which incorporate into nascent viral RNA chains, causing their premature termination. We'll see the detailed mechanism later. Anyway, this drug was developed by the Gilead Science as well as a treatment for Ebola virus disease first. Remdesivir was rapidly pushed through clinical trials due to the West Ebola virus epidemic of 2013 to 2016. It was eventually used in human patients despite its early development stage at that time. Actually, in case of Ebola, it was significantly less effective than other monoclonal antibody treatment. However, the trials established a safety profile for human beings. And moreover, laboratory tests show remdesivir is effective against a wide range of viruses, including the single-stranded RNA virus like coronavirus, the SARS coronavirus, or MERS coronavirus. Based on previous researchers on Ebola and coronaviruses, the effectiveness and safety of the drug have been proven. With this basis, the drug could be available for compassionate use, which means remdesivir is provided for treatment of COVID-19 patient without participation in a formal clinical trial. I mean, it can be used without official approval by FDA. Then how does it work? Let me see the detailed mechanism from now on. Remdesivir is it shares the similar mechanism like the soposbuvir. Remdesivir is also prodrug itself. It is still in active form. And once it is administered into the human body, it is metabolized as active, we call GS441524 hard, a kind of adenosine nucleotide analog. Look at the structure, it's similar. And it can interfere with the action of viral RNA polymerase. We call this specifically RNA-dependent RNA polymerase because the genetic material of the virus is RNA. The template is RNA, and with this RNA template, the polymerase is gonna make RNA, the newly synthesized RNA. So we call it RNA-dependent RNA polymerase. And it prevents the proofreading of viral exonuclease enzyme, which means it doesn't repair by this enzyme. The more detail, here is more detailed viral, the coronavirus cell cycle. As you know, the virus is attached to the host cell surface and enter injecting their genetic material, in this case, RNA, specifically positive sense RNA. A positive sense means same polarity of mRNA. Therefore, the positive sense RNA can be translated by host ribosome, generating very important protein here, RNA-dependent RNA polymerase. They have information of RNA-dependent RNA polymerase into their own genetic information. And RDRP directly mediate the synthesis of negative sense RNA. If you transcript or replicate the positive sense RNA, the negative sense RNA is produced. So it acts as template of various distance positive sense RNA. If RDRP replicate negative sense RNA template, it produces another positive sense RNA, which is going to become genetic material of virus of next generation. And then if RDRP transcribe the negative sense RNA template, it produces various RNA fragment, mRNA fragment, which is going to be translated into many viral proteins like this. Now you see how important RDRP activity is. However, once remdesivir incorporated with RDRP, the activity is disrupted. As in the following three cases, the antiviral drugs work by providing mutation on RNA chain itself, or stopping the replication process, or backtracking the RNA chain. We don't know what is exact mechanism yet, but in any case, they can inhibit the proliferation of the virus by disrupting the viral RNA replication or transcription. And I told you before, this drug can protect from the repairing by exonuclease. It might happen because RNA containing remdesivir take on a strange shape that does not fit into those kind of enzyme. To find out for certain, they would need to collect more structural data. Finally, I want to show you how much it is effective with some research results. Here are antiviral activities of some drugs against SARS coronavirus 2 in vitro condition. The left and right y-axis of those graphs represent mean % inhibition of viral yield and cytotoxicity of the drugs respectively. Even though most of them are working, you can see remdesivir is highly effective in the control of SARS coronavirus 2. So now I hope that remdesivir save patients suffering from the novel coronavirus disease. Thank you.
Okay, excellent. Question to Sejin. Sejin, don't close it yet.
Actually, I cannot understand the positive and negative RDRP proteins. Can you explain one more time to us?
Okay, so actually, you know, the nucleic acid has a polarity 5' to 3'. If in the case of DNA, one template has positive sense, the other has negative sense. In this case, they have RNA as a genomic information, a genomic structure. They have, if the genomic material can be transcribed by host ribosome, it is positive sense RNA. If not, it is negative sense RNA. And positive sense RNA and negative sense RNA has backward polarity. So if you replicate positive sense RNA, here is 5', here is 3', and the other negative sense RNA is 5' here, 3' here, and they have their own information. So it is complementary strand having the backward polarity.
Okay, for now, let's just keep that in mind. Positive strand and negative strand. We will handle this in 2 weeks. Okay, not next week. The next week we are going to talk about this in details. Just keep in mind, you know, keep that curiosity. What is the positive and negative kind of a meaning? Okay. Other questions?
What is backtracking?
Actually, the enzyme move backward from the in the direction. I mean, the RNA polymerase move from 5' to 3', but if backtracking happen, they move backward from 3' to 5'. It is kind of error.
Oh, and that's occurred by remdesivir.
It can be. It is one possibility, but we don't know which mechanism is happened by remdesivir. These 3 are possible case.
So in this case, the red color is the drug. So the compound can be incorporated into here, the double helical structure, right? Now, it can block moving on to the synthesis side, or maybe replication side, or maybe checking side. Once abnormal structure is incorporated into the double strand DNA or RNA, they may have some problem later. Okay, that problem can happen on the synthesis step or checking step or different kind of steps. That is not exactly known which step is the most important, but you know, unnatural structure is incorporated, then something bad can happen to the virus.
Okay, thank you.
Can you move to the next page? I saw something. Okay, here. We call the compound activity as IC50, inhibition concentration 50. Okay. Now, at least I wish you guys can understand what is the meaning of this the graph. Now, if you look at those x-axis, it's an increase of a compound concentration in log scale. You can see remdesivir, it has a 0.1, 1, 10, 100. It's increasing micromolar concentrations. Now, if you look at those y-axis, it's a percent inhibition. If you add more compound, then you see 100% inhibition. If you add a low amount, it is a 0% inhibition, right? Now, if you look at those 50, there's the dot line that is 50% inhibition. Then you can look at those concentration. By looking at this, the graph, remdesivir, what is the IC50 concentration? Approximately?
1 micromolar.
About 1 micromolar, right? Then let's look at the next one. Chloroquine, and that one has approximately?
1 micromolar.
It's a similar, activity-wise it's a similar, but it does not have a sharp increase. The next one, favipiravir,
50.
it's about 50 or 70 or something like that, right? Now, 1 micromolar versus 70 micromolar, which one is more active?
1 micromolar.
1 micromolar is more active. Even though we use smaller concentration, we can block the function half, okay? That's why this compound is more active. Understand?
Yes.
So this is how you read those IC50 values. So we'll come back to this kind of the example later. So for now, let's move on to the next one. Okay, last one is by Dongwoo. We have seen 2 Gilead the compound. One is recommended for coronavirus, originally developed for, Sejin, would you take out your slide? Now recommended for coronavirus treatment. There is another compound which is offered by Japanese government, and Dongwoo will talk about it.
Okay, so I'll share my PPT. Oh, well, actually, when Sejin had talked about the 6 drugs and its individual toxicity and inhibition and the 6 graphs that we just talked about, the one drug that I'm going to talk about now was in the graph. So that was called favipiravir. I just saw that it doesn't have any effects compared to remdesivir, so I'm quite feeling sad, but still I'm going to present my presentation. The drug I'm going to tell you about is Avigan, and Avigan by Japanese words. So as you can see, it's a drug developed by a Japanese company. So the table of contents is shown in here, specified into 6 categories, but I'll try to do it as fast as I can and also comprehensive. So you may have or may not have heard about Avigan, the everyday use. If you're interested in searching various things in YouTube, you will know about an offer that Japan has given to other countries that they were going to give government-insured pharmaceuticals called favipiravir or Avigan to other countries who are suffering from COVID-19. And also, if you search about Avigan in the YouTube in Korean, you will hear these attractive-looking videos that are telling that Avigan is kind of dangerous or somewhat similar to that. So there are various opinions coming out from different media platforms, and we don't know if this is fake news or not. So I will tell you about the main facts of it and how it's going to affect the real science committee, though we don't know the actual mechanism and the actual effectiveness, not yet. So Avigan, as known as favipiravir, or also known as T705, is an antiviral drug developed by Fujifilm Group of Japan with activity against many RNA viruses, especially in influenza treatment. So it's available in both oral and intravenous methods, which means to stick a syringe in your waist, and you can inject it in by that method too. So it was developed during chemical screening tests, which is the original method of developing medicine, which is to just shoot in thousands of chemicals and test if they're effective or not, and they just luckily found this. So this Avigan is a pyrazinamide derivative, which is a chemical which is used to treat tuberculosis. And it is also a prodrug, which we have talked about in the two previous drugs. So its phosphate structure is also protected by other chemical groups, and the chemical groups are digested and taken into use after it's being metabolized. So in experiments, they have shown effectiveness not only in influenza viruses, but also in the West Nile viruses and the yellow fever, and also other types of things. So we want to know why it has so much various effects on different kinds of viruses. So as we talked about in the before, Avigan is also an analog of nucleosides. It selectively inhibits the RNA-dependent RNA polymerase, RDRP. And the mechanism, and the picture of RDRP is shown in the left, and the mechanism is shown in the right. It's really an easy diagram showing that it stops, marked by an X, the propagation of the virus inside the host cell. So the favipiravir, the original molecule, is shown in here, and when it got incorporated inside the host cell, it got phosphorybosolated and phosphorylated by various enzymes. And this enzyme is called IMPDH, which was found by research papers many years ago. And this inhibits the replication of the protein synthesis of the virus cell. So we could, no, not the protein synthesis, but the replication. So this is a very important diagram showing the mechanism of favipiravir. And the same research paper shows that T705 also inhibits viral replication without influencing the synthesis of cellular DNA or RNA, so which means that it will not have any harmful effects on original cells, except the virus. So this is actually some data that I've archived from this research paper, and the previous presentation by Sejin, we have known that remdesivir is an analog of adenosine. However, this drug, Avigan, is actually recognized by both guanosine as an adenine analog. So it has an ambiguous base pairing by the polymerase. But nevertheless, it doesn't deter the effect of inhibiting the replication of RDRP, so it's actually a very useful chemical in both terms. So, but if this is very effective, we want to know why this is being inhibited and precariously prescribed in countries all over the world. So the drug is actually supplied only by the discretion of Japan's Health, Labor, and Welfare Ministry, or the Fusengdodongsan. So it is only manufactured upon request by the Japanese government, and it is not available in pharmaceutical companies or overseas. So we might as well not see Avigan in our daily pharmaceutical stores around our homes. And it is also used only when there is an outbreak of novel influenza virus infections, which are not effective to any influenza antiviral drugs that are known before. So we want to know why, the reason of this. So this is basically because two reasons are behind this reason. And the first reason is that there are only 4 FDA-approved influenza antiviral drugs that is currently being known by the FDA. So that means that there's something's wrong with this drug. And the problem with this is that the animal experiments that which showed the potential for teratogenic effects on fetuses, so which this means that pregnant women which have been prescribed by this drug may have negative functions while they are doing their growth of their baby. So actually in 2014, if you see this news, I know every one of you won't read this newspaper, but it's basically saying that the development for this drug are saying that they're very unfortunate because these effects have been occurred. However, as the current pandemic has coming to rise, Avigan is being variously tested for if it is really effective in treating the disease, nevertheless of the effects. So in March 2020, approval has made in China with the name of favipiravir to treat the influenza disease with this drug. And also it was approved for clinical trials for treating the COVID disease. And after that, as a week passed by, Italy has also approved the drug for experimental use and have been conducting trials in 3 regions most affected by the disease. So governments are actually being very optimistic and hoping that this drug will be effective in curing COVID-19 as well as other many drugs. So before I end my presentation, I want to inform you guys about that there are very immense tests being performed to test if it is really effective or not, and political disputes which are causing very much havoc in our country and the other side of the borders are only causing negative problems. And we have to focus on the scientific parts of it to realize what's good or what's not, and really need to know what's effective for our humanity to survive. So this is the end of my presentation. And thank you very much.
Okay, thank you very much, Dongwoo. So in Dongwoo's presentation, we have seen the molecule is also nucleoside of the mimetic compound, very similar, but it's much smaller, and also it doesn't have a sugar motif itself. So maybe cell penetration is not a problem, but it may have a potential toxicity. So we'll see whether that compound is really useful or not. Dongwoo, would you allow me to, would you take out the your slide? I wanted to put my slide. Okay, thank you. Okay, so almost we are out of time for today. So let me finish today's. So there are several drugs like the Avigan or the other compounds which has already tested the compound for other disease. As said, in that case, we don't need to get FDA approval. Partially true, partially still, we need more tests. Now, at least clinical trial. The clinical trial phase 1 is for safety test. So safety is the most important, even though it doesn't have effect, at least it should not be dangerous. So this class of compounds, once they are tested for other drugs, their basic safety test has passed. That's why that part we can skip. But next step, clinical trial 2 and 3, for the efficacy test, activity test, still we need to confirm and go for FDA approval process, okay? But some of these molecules, this molecule I'm showing you now, ivermectin, is also in newspaper right now. This molecule has been developed a long time ago for parasite treatment, you may have heard that some of the 구충제 may have some effect. So this kind of old drugs, which has been used for other purpose, now if we are in urgent cases, we can retest all the commercialized drugs for new purpose for against the coronavirus. That makes sense, right? In that case, at least we know those drugs are not very toxic, and they may have some chance to show new activity against the new enemy. So this kind of a process we call drug repositioning, putting reposition for the drugs. That's what that means. Okay, before I close the class, so I remind you again, now it's the time you start reading this book. If you don't have this book yet, then order it right away. In 2 weeks, I will give you the assignment based on this the book. So start reading this book, okay? Now, next class will be next Tuesday. And then the next week, the topic is cell structure. So we need to understand our own cell's structure and also bacteria and virus, that we figure out those differences a little more in details. Especially for this week, the structure of cells, there are beautiful movies. Don't just take just as a movies. Those beautiful movies has very good meaning why they described that way, okay? Every movement of those cell organelles, they have specific meanings. So try to understand which part you are looking at and what they are doing and what's the connection, what's the function of them, okay? Every piece of those movies has a very good meaning, very well-designed movies. So I want to encourage you to think very deeply when you look at those movies. Same way, the quiz deadline will be next Tuesday. And also there ups, this assignment 5. Assignment 5, the due date is the same as a Wednesday. Same approach we are going to take. You will choose, I gave you some specific names you can choose. And then you choose one of them. And again, first come, first served the approach. So you pick up one. Then the later comers, you should choose the different topics. I put maybe around the 10 topics. You can choose what you want. And last topic, last choice is you choose your own more interesting topic on your own. Then that's also possible. Okay. So if you don't have urgent the question, then we finish today's lecture. Any urgent question? If not, goodbye for now. And if you have a question, then you may stay and give me a question.
Thank you.
Okay. See you next week.
Thank you.
Thank you.
Bye.
Thank you.
Thank you very much.